it's nice to be presenting something, because I retired in February just before COVID, so I haven't had to do much teaching during this pandemic. So I'm going to try and sort of talk to you about something I've called the third dimension. And whilst we've been involved in this in a sort of medical education domain, I wanted to, for this audience of general biologists, I want to sort of illustrate that there is all sorts of other possibilities. So it's not going to do my slide advance the way I normally do it. Okay, so first of all, just to do the ethical thing, to disclose that we do, the university and the lab that I was formerly director of, do have relationships with several companies now to commercialize some of the products that I'll be talking about. So what I'll break the talk up into these headings. I'm going to start with something about how we've been searching since the Renaissance for ways how to illustrate the depth and three-dimensional form of human anatomy. And I recently wrote a review about this, so it's quite fresh in my mind, and it's an interesting way to start the topic. And then I'll talk about how at Monash University we got into 3D printing for normal anatomy replicas. And then I'll tell the story about how after normal anatomy we did some other things like fetal and pathological things. And then some of the examples of projects we've been involved in that link up with surgical and clinical practice. And then I'll show you examples of how I think it could be used in general biology. So I got asked to speak at the Royal College of Surgeons back in May about art and anatomy in the Renaissance, which is not an area that I'm an expert in, but it did force me to go back and read a lot and to think about how artists sort of at the start of the 15th century, at the end of the 15th century and the start of the 16th, it was really changing over to people like Leonardo da Vinci and Michelangelo trying to use drawing to illustrate three-dimensional form of what had been regarded or illustrated in medicine, which is the far left-hand image, which goes back to Galen and Hippocrates. But these artists like Michelangelo and Leonardo tried to sort of get depth and perspective into their drawings. And they were artists who studied anatomy and did dissect bodies, but it was really Vesalius as an anatomist who got other artists to illustrate, and you can see a couple of his illustrations there. He's considered the father of anatomy. So, and of course we all know Leonardo's famous David, and to sculpt in 3 dimensions like that, to capture that third dimension, requires an intricate knowledge of anatomy. And clearly this is not an iconic figure for nothing. This is incredible knowledge of human surface anatomy. And round about that time, people were creating these things called ricochets, which is basically a flayed, it means a flayed person, a flayed body, where you can see the musculature. And they've been used for teaching anatomy. Here's an illustration of William Hunter, a famous Scottish anatomist, teaching using surface anatomy, and an ricochet teaching some medical doctors back in Hunter's day. So capturing that third dimension has been done in art and sculpture. So as teachers of anatomy, we think about this a lot. But let me take you back to our problem and how this all came about. And it was really a lot of serendipity. So let's think about if you were running, managing an anatomy lab, and some of you are at institutions that probably have anatomy facilities. There's enormous costs involved in running a lab. You need specialized staff. You generally have to have a body donor program, which needs administration and is costly. Some countries don't, for cultural and religious reasons, don't have body programs. So they don't study real anatomy. They don't dissect for that reason. And even in countries like the UK and Australia, you need a certified lab to actually be dissecting a human cadaver. And you all know the story of working here, the Scottish body snatchers that were keeping Edinburgh University going for many years. So that's when everything's been legalised in most developed countries. But there's also an increasing awareness now of formaldehyde as an irritant and also as a carcinogen. And WorkSafe Australia is going to be changing its regulations soon to exposure levels going from 1 part per million, which is really low, to 0.1 part per million, which is almost undetectable, because students standing over the body get exposed to quite a lot of formalin in a traditional lab. And the other thing about having bodies is that some countries have had scandals. The USA had various scandals, and if you look up the BBC, you'll see some of the things about bodies having parts taken out of them and replaced with broom handles and so on. Terrible, terrible things. So when I went to Monash in 2010, I came from a very good department in Western Australia. But when I came to Monash, this is what I was presented with within weeks of taking over. This is supposed to be an anatomy lab. These are pots of bodies with bits of human in them, some imaging, and students just milling around, carrying a white coat. It's like not wearing your mask, I suppose, in public. This was just appalling. So I rushed upstairs, grabbed my camera and took these pictures. And I decided I was going to change things because this was unacceptable to have a wet anatomy lab running like this. We did have a dissection room as well, but we had to buy our cadavers from Melbourne Uni. So we had a very poor collection of stored specimens. They were all in pots, and most of those things on those benches there were actually pathology specimens. So I started to think about the alternatives that were current at the time, back about 10, 11 years ago, which was plastination. And some people might be familiar with this crazy guy here, Gunther Von Hagen, who does these body world displays. He's very controversial, and when he started doing this process, there's a lot of questions about where he got the bodies, because most of them are quite young looking. Anyway, it turns out it's a really expensive process and quite a dangerous process because of the amount of solvents. So I was thinking about how do we get lots of specimens for teaching without having to buy lots of bodies from Uni Melbourne at $10,000 a pop, but get enough specimens for our large classes, because we had 320 med students a year, plus 100 graduate entrants, plus 400 or 500 biomedical science students, physiotherapy students, radiography students, and yet what we had in terms of teaching materials was not good. So what other alternatives are there? Plastic models. Well, here's plastic models. They're inexpensive, but they're 3D cartoons, and they're all right for maybe teaching at high school or something, but not at the university level. So, like a lot of good things, it happened over a cup of coffee. So I was explaining to this chap, Colin, who had come to work with me recently back then, and he, in his interview, he put this down on the table. And I was bemoaning the issues of trying to get enough specimens to teach classes. And Colin said, oh, we should just 3D print everything. And it was one of those flippant comments that I could have just passed over and we could have forgotten about it. But I did know that he was 3D printing some of this type of stuff for his research. And I thought, okay. And Michelle, this lady here, worked for him at the time as an RA. And this chap, Justin, joined us about a year or so later. And anyway, I said to Colin, well, how do we do that? And one thing led to another. So we went out and bought a 3D printer, and we started learning about the technology of how to prepare 3D prints. So basically, and this is not a secret, we published this, and it's very highly cited, which is really interesting, just from someone who's been doing science for all his life. So it's not a secret. This is something that we couldn't patent as such. So all of these separate items are all available. It's just I don't think anybody had thought of putting them together. So what we do is we take a pro-section or a prepared dissection, we CT scan it or surface scan it using an Artex Spider scanner. We sometimes digitally segment out the elements within that specimen, like the bones and nerves and muscles and so on. We use software called Aviso for modeling. And then we used a thing called 3D Co to color the constituents in standard colours like muscles brown, nerves yellow, veins blue, etc. So, and these are the printers that we use, but I can come back to that. If anyone's interested in the technical things, I can come back to that in more detail. And this is our first print that came off. We bought a $100,000 Z-Clock printer, which is a powder-based printer that prints one layer at a time. Each layer is 100 microns thick. And so the first time we sent the item off to the printer, we left it overnight and came back the next morning, and there was a big solid bed of white powder, and we thought, oh shit, it didn't work. But in actual fact, what you do is you take the white powder away, and then nested inside it, once we cleaned it up, this was what we found. This was the very first print we made, and this is the specimen that it's based on. So you can see the accuracy of the nerves and the vessels and the tendons and the muscles. It's extremely accurate. So we called this the hand that shook the world, because to us this was just momentous. So why did we make them coloured? Here's one of our first attempts that illustrates why we did that. Because if you print it in cadaver colours, it just looks like the original specimen. And I sort of thought, well, we're trying to educate these students. We're trying to teach them about human anatomy. Look, we may be familiar with looking at cadaver colours, but the students aren't. So let's make it easier by colour coding things. So we did that. Working with 3D data, this is a 3D data of one of the specimens we had. You turn it into 3D render like that, and then you artificially colour it, and this is done in that software I mentioned. And then eventually you 3D print it, you put it in a good 3D printer. Now, back then, that printer bed wasn't big enough, so you can see there's a join here, and we had to print this in 2 components. So this would come out of the printer in over 2 days, and that takes, in the new printer we've got, that takes about 25 hours, I think, to print in the new UV curable resin printer we have. And this software allows us to change the faces if there are faces in the specimens, so there's not like any patient confidentiality things. And you can see from these that this is not like a plastic model. This is not a caricature. These are extremely delicate anatomical structures, really accurate, and this is exactly based on a real specimen. So we call them 3D printed replicas and not 3D printed models, because they're really a replica of the real thing. And you know what? If we want more for a class, I don't have to dissect another 10 pelvises and store them and bring them out each time, because I would need to buy 10 bodies to get these pelvises, and I would need to pay prosectors to dissect them, and it would take months and months and months and months to do that. If we want a new pelvis, we print it and it's ready the next day. So here you can see where we had an old printer which wasn't able to print a whole torso, but our new printer actually can print a whole torso in one hit. And we had this first series that we did had a total of 57 specimens in it, every part of the human body. We didn't print solid organs that don't have much 3-dimensional form, like livers or spleens, because, you know, a plastic model does the same thing essentially. There's no topography, no intricate detail such as in these specimens. So we didn't print things that you could basically create a plastic cast of. These are printed because they're complex 3-dimensional forms. Printing allows you to change the size of things. A couple of examples here. You can even print out whole torso and hold it in your hand, but this is actually anatomically perfectly accurate. This is based on a real specimen, and it's a beautiful dissection that would take probably a good anatomist probably take them a year or more to do. So we were able to repurpose these old dissections. And the other thing that we could do with 3D printing is we can take other types of data. So this is CT angiography, and you can't dissect this. This is the arterial and venous vasculature of the brain. You can print some bits of it, like circle of Willis, the major arteries, and you can put in more. You can put in less bone, more bone. And things like tractography, we can also print that. And that's a new area that we're moving into. So things that could never be dissected in a million years, you can actually print if you've got the right kind of data. I'll just throw this in as an example. People might be saying, well, we've got anatomy facilities, we've got real cadavers, the students prefer cadavers. Well, I'll show you an example of where there is a situation where students don't have access to cadavers. This is a couple of pictures from a trip I did to Liberia in West Africa in late 2015. They have no anatomy staff, they have virtually no staff at all. They had no cadavers, they didn't flush toilets, they had nothing, absolutely nothing. Basic lecture theatre with poor electricity and light. And anyway, I went out as part of a group from University of Massachusetts and Boston Children's Hospital, and I donated an example of every single one of the prints that we had done at the time. And it was really these moments here that just absolutely brought it home to me, just what we had done. We had created something that students who don't have bodies to study from could actually study proper human anatomy in detail. And we've just recently sent a whole bunch of donations to Fiji and PNG. But if you're natural, you would be thinking, well, they're not as good as the real thing, surely. So we thought we'd test that. So we did a randomized controlled trial where we split students into 3 groups. We gave one group 3D prints and cadaver specimens. We gave one group 3D prints alone, and we gave one group cadaver specimens only. And we did pre-test and post-test. And here are the things that we studied: cardiovasculature. So this was a program looking at the heart. They did a test, and this is the average change in score. And you can see that the 3D print group, the score increased 18% compared to 7 or 8% on the ones that used the other specimens. So this was not what we expected. We thought we'd maybe get parity, but we didn't think it would actually be better. And in retrospect, I wish we had videoed it, because I think the difference is they will pick these specimens up. They tend to be apprehensive about picking up wet, real hearts, human hearts. So that article has been very well cited, because it proves that if you're trying to learn something like human anatomy in this context and you don't have cadavers, then 3D prints actually outperform cadavers. So that was quite a significant finding. So once we'd done the normal anatomy, we had other stuff in jars, including these collections of human fetuses. And so we scanned all of these, and I've recently had these respectfully cremated and the ashes scattered in a garden of remembrance, because we don't know the provenance of these specimens. We don't know how they came to be in the possession of the anatomy department. Most of them were probably 60, 70 years old. And in light of that, we did not commercialize this series, but we do let students use it for the purposes of education, because here you can see a multi-material print where we've segmented out all the bones, the ossified bones, and it's just beautiful for teaching ossification centers in the skeleton, the human fetus. It's just fantastic. Beautiful specimens, and it was just a shame that we didn't know where they came from. Otherwise, we could have possibly let more people in the world have exposure to this. Then we looked, and I had a 1,600 pathology specimens. So I was ready to chuck them all out. Then I thought, oh, maybe we should save some. Anyway, long story short, we have created a pathology series, and we've just finished that, and that's going to be marketed by a German company. So we used for this a combination of radiographic data and RTEC scanning to get the color detail and color form, this 3rd dimension, in better resolution. And you'll see here are some of the specimens. So there's kidney stones, gallbladder stones, stones in the ureter. This is an example of a very rare pathology called tetralogy of Fallot, where there's a swapping of the major vessels. So that, you know, this has come out of a kiddie's heart that died at birth, and you know, it's a unique specimen. We can print it, and anybody can have one. It's a lovely aortic aneurysm, maybe not lovely for the person who had it, but so these are, we think this is a fantastic opportunity because not everybody has pathology specimens. So we're hoping that people will find these very useful. And more recently, we've just gone back to using a combination of real colour and slightly augmenting the colours. These are slightly augmented colours here to make them more cadaver-like. Just so that students kind of can get the feeling that it is a real cadaver. Maybe the brightly coloured ones felt too artificial to them. We've no evidence of that, but I just have a gut instinct that that may be part of the case. When we got started, people heard what we're doing, and we had lots of collaboration with surgeons. Here's someone with a facial tumour, so the surgeon, we can print that for the surgeon. He can see the extent of that tumour before he goes in and removes it. Here's a cyst in a baby's gallbladder system that was printed before surgery and was useful for the surgeon. Here's a broken wrist displaced radius. So we had quite a few clinicians come and get our help. Here's an example of kiddies who have had some medical imaging who have had cleft palate and cleft lip, and we were able to pull up these data sets, turn them into 3D form and print them. And we even tried to print them for replicas for surgery. Other things, like we've had students, honour students, do projects such as this one where we created, modelled in 3 dimensions, created aneurysms in the internal carotid artery and its major branches, and we were putting that inside artificial brains created from 3D printed molds and making casts of a gelatin-like brain so that surgeons could practice clipping these aneurysms. So lots and lots of applications in medicine. Here's another one where we created a paediatric airway simulator model by actually using proper data from babies rather than just making it up as a sort of 3D caricature. Anything that's in 3 dimensions, here's a couple of molecular ones, some receptors, some viruses, anything that's 3D data can be printed. Here's some data from my own work in the laboratory, looking at macrophages and dendritic cells in the retina, microglia, and anything that's 3D, we can print it. And I think this is another huge potential area that people haven't realised yet, that 3D printing could help. Because showing that to a student is really quite powerful to show them how cells interact with nerves. And here's another application might be more relevant to this group. I've got an example of that here in front of me. I'll show you it when I turn the screen share off. Here's a rainbow bee eater, that's a real thing, and that's a 3D print. This is a hooded plover that I printed. I printed the eggs for conservation education down in the Mornington Peninsula. And I used to be a bird egg collector when I was a young boy, and I have some antique egg collections, and one of these is a 3D print, and it's an osprey. It's this egg here. So you can see the potential is enormous, and we recently got asked to 3D print a copy of this beast here, the Tasmaniosaurus, which is a 260 million year old fossil reptile. It's not a dinosaur, I've been told. And that's in display in the museum in Hobart. So huge potential for archiving biological specimens in museums and collections. And I think that some of the museums around the world have started to do that. So in conclusion, if something is 3-dimensional, if it has a 3rd dimension, then it's now possible to capture that information and print it. And as I say, it's probably critical to archiving scientific specimens all around the world. I think we've got a huge potential to replace teaching specimens in biology classes, and examples are where students used to dissect, fixed rats or frogs. I think that could all be replaced. In a way, this 3D printing, is it the new taxidermy? I always wanted to be a taxidermist when I was a kid. Maybe this is how I've ended up being a taxidermist, but without actually doing the stuffing part. And really, it could bring any rare specimens to anyone at any time, human or non-human. You don't need a wet lab. And one of the things I'm not talking about today, but I can ask if anybody's got a question, is we've taken all of this data and we're now launching it into virtual reality and augmented reality environments with another company in Colorado. So data is data, it's three-dimensional data, and we can take it wherever we like. So these are the staff. This is Justin, who joined us. Colin moved on to another job. John Bertram was the head at the time and supported us entirely. Michelle and Lucy are the two technicians that run the lab. And the university, the faculty and the main university were fantastic at funding this. I never wrote one single grant to do this. Just to give you an idea, our last printer cost $230,000, and the university was fully supportive because this stuff does well, it sells, and we get royalties, and it's paid for itself easily. So that's where I'll end the formal part, and I'll finish up with a picture of my favourite beach in Scotland.